Start talking about cellular respiration first. And in order to do that, even for photosynthesis, you need to understand this a couple of concepts. One is the oxidation reduction reactions, or otherwise known as redox. An oxidation is where an atom or a molecule loses an electron or more than one, and a reduction is the gain of electron, and usually these things go together, hence the name redox, because you can't have a loss of an electron unless it has some place to go. So something had to gain it. So usually that's kind of how it goes. Now you're going to see that the electron flow from one molecule to another, in other words, the loss of from one molecule and the gain to the next one, and then it's lost and gained to another one, etc., down a line, is going to be something that can be used for doing different processes. And there's some really interesting things that can happen with electron flow. So let's just talk a little bit more about this concept of oxidation reduction. All right, there's the squeaker problem. All right, so anyway, this is a picture, let's see if I can get close enough here, of sodium and the making of salt, right? And so the idea here is that in sodium, I have an extra electron on the outside. This is one we've already drawn out before. And this is chlorine. It's going to take up that additional one. When that happens, this becomes positively charged ion, correct? And this is going to be a negatively charged ion, and an ionic bond may form, but this was an oxidation reduction reaction. So I will show you it drawn out. That is, this is one of your handouts, by the way. That is, if I have sodium and it becomes a positively charged ion, this is what we call an oxidation. In other words, it lost an electron. And chlorine, because it became a negatively charged ion, it took on that electron. It was a reduction. Okay, so this is an oxidation reduction reaction. If you are something that takes on the negative ion, all right, so if you get reduced, then you are the oxidizing agent, all right? And if you are the one that is donating the electron, like in this case, that is, they get oxidized, then you are the reducing agent, because you're going to be reducing the thing you're next to. Okay, so those terms can be a little bit confusing. All right, so just so you know. Another form of oxidation reduction, here I have a bigger one of these. This is on your handout, but this is a little bit better maybe, is just the reconfiguration of electrons. And this is, as we go through some of the processes, you're going to see that in some cases, the molecules seem to have more energy or the same amount of energy, even though they've lost some parts to them. Like, in other words, lost a carbon, but they still have a lot of energy, and you're like, how could that happen? Well, basically, it's a redox thing. And I'm not going to go into why that is in each case or anything, but I just wanted you to kind of get the idea. And that is, an oxidation, remember, is a loss of electrons. That, in this case, the central atom's electrons are further away from that central core, and that's a loss of electrons. And in this case, this is a reduction. That is a gain of electrons. The central atom, in this case it's the oxygen, has the electrons closer around its central core. So that's also a form of oxidation reduction, which maybe isn't apparent. Another thing that you need to know is something that we call substrate level phosphorylation. Now this happens in some cases where an enzyme is going to take some substrate. So in this case, it's this molecule called PEP, and later I'll show you what that is. And it's going to take a phosphate group off of that, and it's going to phosphorylate ADP, or adenosine diphosphate, which, remember, is a cyclic nucleotide. That cyclic nucleotide will take on that phosphate group, and become ATP, which is our energy carrier molecule throughout the cell. And it leaves a byproduct of what we call pyruvate. Now in this case, ATP would be gained from this. It's an enzyme that is taking the phosphate group from one substrate and passing it to ADP, making ATP. This is called substrate level. Now this is opposed to the other way of making ATP, which is going to be called the electron transport chain that we will talk about later. So you just need to know what that meant. And then the last thing I wanted to point out before we start the process is this is also another type of cyclic nucleotide. This is what we call NADH, and it's NADH at the end. But NAD will take on electrons, and as it takes on electrons, these will be taken on in their excited state. So they'll be gleaned off a molecule that had a higher energy level, and it's going to keep those electrons at a high energy level and then have it in this molecule, NADH, where it can then donate it. In other words, it can become oxidized in that high energy level. And this is going to be an important thing. So these are kind of like electron carrier molecules. I think I mentioned this to you when we were talking about nucleotides or nucleic acids, that there were some that carried electrons, and this is one that would carry electrons. Now notice also that they add hydrogens to the molecule. I always think of this as kind of like an end cap, you know, like you add the electrons and then you add the hydrogens on to kind of keep them on. And then when you take the electrons off, the hydrogens are going to be released. So you'll see that that happens fairly frequently throughout the process, and just so you know what that means. So this is a molecule called NADH, or NAD, that will become NADH, carrying the electrons. There's other ones. There's FADH2, and there's in photosynthesis, there's NADPH, which is a very similar kind of molecule. So we will be talking about these as we go through the process.